Magda Kazmierska had worked with very lonely elders in this community center and helped them to put together a dance that they did in front of all of us. It was just extremely moving. And Sergio said, we don't start with research. We start with engaging with these communities. That's where this all begins. And then research will come. And I totally got it. I think I got it for the first time. Elena is working with Kate Poseen, Victor, a number of other of you, thinking about psychometric ecological validity of neuropsychological assessment in diverse adults. She's thinking about Africa, South America, amazing program she's developed. Tanisha is already clearly an innovator, you know, someone who is one step ahead, or maybe 2 or 3 steps ahead of the rest of the world. What we don't see, we assume can't be. Black women's experience of healthcare and possible brain health futures using Afrofuturism and digital storytelling. She's our 2024 awardee, Elena 2023. Then we'll have a little bit of time for discussion and closing. So far, I think pretty appropriate. Sergio was our first scholar, and wow, what's happened in those 5 years, creating outreach programs for Spanish-speaking adults. Alyssa Bernstein, GBHI fellow in our first class, still working with Kate and others, adapting dementia diagnostics for primary care physicians. Boonlee T has the largest, I think, Asian outreach program in the United States, and has done really innovative work, in particular with Mari Lu and the language team, thinking about differences in the Chinese language, what the meaning of that is for cognitive testing. Charles Winden and Salva Spina were co-PIs in 2020. Charles also just an amazing force in our community. He has set up a program in Bayview. He's a magnet for trainees. He's an exemplary role model and is increasing representation of African Americans in our autopsy cohort. Elena, adopting TabCat for diverse population. And again, Tanisha, using Afrofuturism storytelling to empower Black women, disrupt power imbalances, and envision future care models. So we've put $120,000 into these awards. They will continue. I think soon we'll be looking for new applicants for 2025, but we couldn't have better people than Elena, Tanisha. Thank you. So go forward, Sergio.
All right, let's see if my Prezi link works. Yes, okay, great. I'm gonna jump straight into this talk. I have 10 minutes. Just a few words about the outreach program. The idea emerged from a need to be open and communicate to each other around the work that we do in the MAC, facing the community, and also let the community know about what we're doing. That's kind of the root of why this idea began. I'm going to be talking about the work that many individuals are doing across the MAC, just trying to summarize everything so that we're all aware of what we're doing. And these are the major players, but I'm sorry if I missed anybody from this picture. Also want to acknowledge that every year we have Atlantic Fellows that are an integral component to varying degrees in our outreach program. Some come in, come out a few times, others stay throughout the year, and others stay multiple years, like Alex and others that have continued to contribute to our outreach efforts in the Bay Area, even from abroad. I want to take us to a hypothetical scenario. Let's imagine that we are all dermatologists. We're interested in studying the prevalence or the incidence of melanoma. And I'm starting a research study trying to understand melanoma. Well, it's pretty clear what I would need to do, right? I would need to develop an outreach program where I would actually enroll people that span this breadth of skin colors into my program. And I would have to follow them over time and figure out what is the prevalence of melanoma. There's a perfect model behind this, right? We understand that skin color is driven by melanin, and there's a direct relationship between the risk of melanin and incidence of melanoma. And people have done these studies, and they found that there's a wide difference, right? 3% lifetime risk of, oh, excuse me, it should be the other way around. 0.1 lifetime risk of melanoma for dark-skinned individuals and 3% for light-skinned individuals. And there's a very a clear model driving this, right? You have a social identifier, race, which correlates with a very clear biological tag that puts you at risk of a specific health outcome. And that's as simple as the model is. But when it comes to the brain and neurodegenerative diseases, we don't have that luxury. In other words, race, as we identify it in society, is not a reliable proxy of a specific biological marker that then we can tag to understand brain health outcomes. It's just not what happens. In fact, we're dealing with a disease, with a group of diseases and an organ that is highly complex, and there's a bunch of extrinsic and intrinsic factors that coalesce to give specific brain health outcomes of interest, right? We all know the risk factors that we're talking about. Some of these are medical risk factors. Some are behavioral risk factors. And that's where the challenge lies when you're trying to build an outreach program, even because it's not as simple as just recruiting people from different racial groups. But nonetheless, we continue to think, I have to say, many researchers continue to think that there is a biological basis. And it's a matter of, finding the right sample size or the right markers that are going to explain why we see these disparities across different socially defined racial groups, regardless of the fact that the evidence is just pointing us in a completely opposite direction because there's so much overlap between racial ethnic groups. And when people steer away from biology, when they open their eyes to that reality and they at least have curiosity for what else could be going on, they turn to culture and they kind of make the same mistake. You overgeneralize culture, and I'll just stick to Latino culture. This is a concept that I've talked about this multiple times. There is no such thing as a Latino culture. I come from Peru, and in Peru, there are not innumerable, there's many different cultural entities, cultural backgrounds, right? Beliefs, practices, extremely diverse country. So now extend that to all of Latin America and ask yourself, how is it that here in the States, we think of a Latino culture that could help us explain health outcomes? It's absurd. It's absurd. That is not to say that biology and culture don't matter. Of course they matter, but you got to think more of the individual. So when we bring this back to this schematic and we start to understand that there is a relationship between these medical risk factors and these behaviors, there are links obviously between them. Well, you can take things a little bit further down upstream and realize that just like lack of exercise can predispose us to something like obesity, well, there's other things that you may be exposed to that elicit the lack of exercise, right? And that's just the underlying mechanism or the underlying understanding, I think, that is driving our outreach work and our approach to outreach. So this idea that there's upstream factors to keep in mind, midstream factors and downstream factors of interest, and that there isn't a simple model like the dermatology model. And this is very, very eloquently illustrated when you actually interview people in San Francisco and you ask them, what are the challenges around you having the health that you want for yourself, right? There's no culture that doesn't want health. And these are the factors that come out when you do qualitative interviews, the lived experience of people, and not so much directly a racial factor or a specific cultural factor. Fortunately, this is a study from 2020, I believe. I think researchers are increasingly opening their eyes to this, or at least having curiosity and testing these testable hypotheses. This is a very nice study that, independent of racial identity, ethnic identity, they found that neighborhood level factors are a good predictor of overall brain volume and hippocampal volumes, meaning, the environment matters, where you live. It's more about place than race, is the message. So with that awareness in mind, a few years ago, I came up with the idea of developing a map for San Francisco that can show us vulnerability. In red are the most vulnerable neighborhood measured across many variables that the city of San Francisco collects at a neighborhood level, not a zip code level. We correlated that map with a map showing where do people come from to our MAC. Where do people come from to our clinic and our research projects? And you see that the most vulnerable neighborhoods are the least represented and least served. Not perfectly, but you see the pattern there. And not coincidentally, because of the history of this country, you also notice that these are the same neighborhoods that are highly represented by people of color. Okay, so now you have an empirical approach to outreach, which I always keep in mind. And then you can say, well, San Franciscans truly embody a wide array of social and environmental factors that influence their brain. And therefore, we should seek to bring as many people as we can from San Francisco and the Bay Area so we can characterize this diversity and then later understand how that diversity of exposures influences brain health outcomes. That's kind of the dream. So many of you have seen this map. This is the initial map that we created where we said, okay, we have to go to these areas. Let's go to these neighborhoods, make relationships. If they speak Spanish, let's bring people that speak Spanish. If they speak Chinese, let's bring people that speak Chinese, and let's do the work of creating awareness. And like Bruce said earlier, being there to serve, knowing that if we do a good job and we are clear about our mission, people will naturally express interest in wanting to come to our research programs or clinics and so forth. We've extended our footprint beyond the city. And this is a graph that the NIH obviously wants us to always present things according to race, ethnicity. So that's why you're seeing this presented in this manner. We put together this chart for our ADRC renewal, where we listed all of the partners that we've developed across many years. It's a lot of partners. It's impressive when we look at it all together. And in bold, I'm highlighting the partners that are active this year. So there's a lot going on. This is a cool visual that I've been working on for this year. This is basically, don't get bogged down too much in the colors and the lines, but what you're seeing here are, this column is showing what we call our pillars of action. Up here, you have the clinics in purple, the community-based clinics that serve as a referral of cases to our observational and clinical trials. And we're working closely with clinical trials recently to try to increase our pipeline of referrals. Here you have our educational efforts. And if you follow the color, you can see where education is taking place, meaning this column here represents the places where we're interacting with people in the communities that we serve. Senior centers, consulates, clinics, and many other places. So these are the places located in those neighborhoods of interest. These are the offerings. And what I'm showing here is that when it comes to research and clinical care, that's our goal, to incentivize recruitment into clinic and recruitment into research. I can spend a whole 45 minutes just talking about this, but I'm gonna move on. Just to give you a little flavor of the extent, this is just for the academic year of 2023. How many events did we do in person where we bring education around the brain and why it's important to participate in research across these different cohorts, and how many people end up enrolling in research? Every year it's different because every year we tap into different individuals and everybody has their own conceptions of research. We also have a lot of virtual events every year. And also varying success, but we always are able to incentivize enrollment. I say, quite frankly, that it's not difficult at this point to incentivize enrollment. I'll speak for Latinos, but I think it's true for other cohorts as well. Incentivize enrollment in research. Am I going too fast? So increasingly, we're becoming very creative in the ways that we try to bring education and awareness about the brain, something we call brain literacy, into different communities. And the creativity comes from the people, right? The people that join the program. It's not one person coming up with these ideas, obviously, right? So we have a fellow that comes in, and you're going to hear from Tanisha's amazing work. I could have never imagined that something like this could have happened, and until someone like Tanisha comes along and says, "Hey, why don't we do this?" And it turns out to be awesome. And I'm just showing a few examples. This is the event that Bruce mentioned. This was probably our largest arts-based exhibit we did last year. The idea here is that Creative Minds, which is our arts-based program funded by the city, every year we do an exhibit where we showcase the work of all of these older adults, elders that have produced the arts, and they bring their families and they bring their communities into the center. And we use that opportunity to talk about the brain, talk about clinic, talk about research. Excited to say also that Jessica de Leon and her team are becoming a part of the idea of an outreach program. We're sharing the way we acquire data and our general approaches, and she's kicked off this Filipino outreach effort as well. And like I said before, I think just speaking just now for the Latino efforts, I don't think we are at a point right now where it's challenging to bring people in to the MAC. I think the challenge is to bring more people. Meaning, I'm sure that if we do these super large events, and many people are going to express interest, but I can't bring them in the next month, And so that's why we're moving. Our goal, our hope, and we've talked about this for a long time, is to move towards community-based research. Charles, I, and others in the team had a meeting last week with a Bayview Senior Center, and that's where we're heading, to create a hub in Bayview, for example, where we can actually see people on site and bring them to the MAC just to do biomarkers and imaging. I want to end with this project, one of several projects that we do as an outreach community, where, as I said before, we want to be able to characterize the diversity of our research participants, the diversity of lived experience, so we can have more predictor variables that we can use to analyze brain health outcomes. So a few years ago, we started this project of asking people about their lives in ways that we had not done before. We had not been asking about income ranges, for example, in the Memory and Aging Center. So we've incorporated that in many other exposures. I'm proud to say that this questionnaire has been very well received in general across different languages. We have about 250 samples now completed, and our goal is to expand and be able to collect this data from as many people as possible that come to the MAC. I want to do a shout out to Wei Ming, who's been amazing at using this data while we've been collecting it, and she's helped put out more than one publication, posters looking at correlations between early life adversity and cognitive outcomes, controlling for race even and ethnicity. That was fast. I felt like I spoke fast, but I hope I was clear, and thank you all so much, and thank for every PI that is contributing to these efforts. Thank you. Yes. Yes. Tanisha, I think, are you going to touch on that or no? Or Charles, maybe? Charles is a project dedicated. I can't speak data-wise, but maybe you want to say something about this, Charles, or no?
All right. Surgery is always a tough act to follow, and I'll try to get through this. I apologize. I'll probably skip a couple of slides just to make sure we're on time and also that Tanisha has enough time to speak. But thank you everybody for being here. Today, I'll be talking about the psychometric and ecological validity of neuropsychological assessment in diverse or older adults. I don't think I'll spend too much time talking about why this is important because hopefully everybody in this room is sort of aware. But generally speaking, neuropsychological assessment, at least from a neuropsychologist, is a core component of ADHD diagnosis, and one of the things that we know is happening both in high income countries, but especially in low income countries, is that older adults are becoming rapidly more and more diverse. They're diverse across all of the different factors and variables that Sergio was talking about. And I think the unfortunate reality is that neuropsychological assessment is sort of catching up to that diversity because most neuropsychological tests have not been developed. I actually have not before, and now I'm just saying are not. Like even in 2024, most tests are not developed nor validated in diverse cultural groups. And I really like this statistic. I apologize if you heard this before, but I think it's so important for us to be aware of this. Western educated, industrialized, rich and democratic countries or weird countries, the populations of these countries constitute 12% of the global population. Consider this, 12%. However, 96% of peer-reviewed behavioral research is in these countries. 99% of developmental neuroscience studies are done in these countries. So the question is, I think it's kind of a controversial question, but think about how much do we really know about brain development? How much do we really know about behavioral science if we are constantly doing research only in areas that are already so resource rich? And specifically for neuropsychological assessment, a very important thing to consider is that excellent reliability and validity that you establish in a single group does not guarantee that the test's ability, the test is able to provide meaningful data across other different groups. So in this project, we wanted to examine psychometric and ecological validity using traditional and tablet-based cognitive measures that are used in large consortia. So I had two specific aims. First one was to examine potential measurement bias using a measurement and variance approach of the National Alzheimer's Coordinating Center Uniform Dataset Battery, which is our paper and pencil tests, and also several TAP-GAP measures, which are digital measures developed by Dr. Kate Fassine sitting right there, and Dr. Gerald Kramer sitting right there, and a lot of other collaborators at the MAC. And the second aim was to examine the strength of associations of these tests with sort of a validated functional measure like the clinical dementia rating. Because of time, I'm only going to speak about first aim today, but if you want to hear me talk again, please let me know. So in terms of participants, this was actually a massive sample because we included older adults, which we defined as being of age 50 years and above, who were enrolled in observational studies at the UCSF Memory and Aging Center. Those are our three big cohorts, the branch cohort, ADRC, and PPG. We also included data from the Multi-Partner Consortium to expand dementia research in Latin America, or REDLAT, and also the Artful Lefties, I never know how to say it, All FTD Consortium, which is a large multi-site consortium aimed at enrolling participants across the FTLD spectrum. So I was asked to talk about challenges. You're about to hear challenge number one. So challenge number one is participant diversity. So I actually applied to a number of other studies, which I'm not going to mention, that collect longitudinal data in this country and beyond. And two of my requests were denied. One specifically was denied because of lack of sufficient diversity in our sample, to answer your question. So I thought it was just really interesting to see that, on the one hand, I do appreciate it. They did let me know from the get-go. But on the other hand, I think it just, again, raises this awareness that there are multi-site, multi-million studies out there that are still not enrolling diverse participants. So this is just the overall breakdown of the participant population. And you can see that we have a little bit over 2,000 people. They are fairly diverse in terms of educational attainment, age. I think another interesting thing is if you look at the enrollment of female participants, in our MAC studies, it's over 51%, which is actually consistent with the fact that women do live longer and have higher risks for dementia. But in other places, like for example, in the Red Light Consortium, the minority are women. And I think it kind of highlights, this idea of like access to research, access to care, all the burdens that women likely experience even at older age, not being able to have a lot of free time to participate. So you can see the breakdown by being self-identifying as being Hispanic or Latino, and also percent of people who are cognitively impaired. So just a brief review of the battery. You're probably all familiar with these tests, but these tests are typically divided by different domains, which we then use to kind of inform our decision-making about where do we think the areas of impairment are, does this look like a certain phenotype? And if it does, is this phenotype associated with a certain pathology? So these breakdowns are actually used, I developed this model using published data. So, people who do develop the National Alzheimer's Coordinating Center battery, they publish this test as being, Benson figure is actually a test of both visuospatial functions and memory. And so this is the model that we'll be testing in measurement invariance. Other challenges. So challenge number 2 that we experienced was actually, so TAPCAT measures were designed to be sort of very user-friendly, ideally ideally so they can be administered by non-trained personnel. You don't require a trained neuropsychologist, of which there are very few in the world. And we have built in practice trials, meaning that, if somebody can do the practice trial, they get what the test is about, they move on to the test. But if they don't get the practice trial, they don't move on to the test, right? Because we don't want to collect data that might potentially be invalid. Well, what we did find is actually a lot of dementia patients in the red lot sample, patients enrolled through the, in multiple Latin American countries, were failing the test, meaning that we were not getting data in 45% of dementia patients on the flanker test, 26% on set shifting, and 49% on this running dots spatial working memory test. I think this is very important because it helps us as a team develop better practice tests, right? We can think about how do we revamp the practice test so that it provides a little bit more education, a little bit more support, and helps us collect more meaningful data. So the TAPCA team has been working on this. And so now I'll be going through different tests by domain and showing you the slides are going to be very consistent, but in the top part, you'll be seeing distributions in clinically normal participants across different cohorts by color. And then in the bottom part, you can see how many people generally, not just clinically normal participants, are at floor on the test, at ceiling, meaning that, if the test has a range, you'd never want a lot of people to be at floor, meaning that everybody's doing super well, getting 100%, or at floor, meaning everybody's getting 0%, in order to get meaningful data. And then in blue here are the areas under the curve, or the discrimination accuracy between controls and individuals with dementia, but only in the red lot sample. And the reason why I did this only in the red lot samples, because we know that this tests work in the US samples. We've already done this before. I've published on it. A lot of people published on it. So I thought it would be interesting to compare. So for attention tests, you see digit span, we see a fairly okay distribution in normals, same for digits backward, and dot counting also does okay. And these tests don't appear to have particularly problematic floor or ceiling effects. But if you look at the overall diagnostic accuracy, dot counting actually does perform a little bit better compared to our normal digit span forward and digit span backward. I will say, however, that because dot counting was only administered to about half of the sample, that sample was a little bit more, had more education, were more likely to be urban. So I wouldn't interpret this as dot counting being the best measure of attention. So I think that's very important for us to consider. So when it comes to memory tests, we use 3 different memory tests. Craft Story, which is a logical story memory test that was adapted into Spanish and Portuguese. The Benson figure delayed recall, and favorites, which is an associative memory task available on TAPCAT. And unsurprisingly, when we look at individuals at floor, this is a sample that is clinically mixed. There are a lot of patients with dementia, particularly Alzheimer's type dementia. So we do see some floor effects. But it's interesting to see that about 11% are floor on favorites compared to 19%, so 20% on craft story. Very few people are at ceiling. And I think another important thing to notice that you're probably noticing here is that on craft story, even these people are cognitively normal. There's nothing wrong with them. They were deemed by a multidisciplinary team to be healthy, but you can see that the performance is very low. So again, we cannot really make any conclusions as to why, but I think it's important for us to know that we are collecting data that we want to be meaningful. And if the data are not meaningful, it creates a problem. And overall, the tests are performing fairly well in terms of discrimination. Favorites in particular has an AUC of over 0.92, which is fantastic. My favorite topic, speed and executive tests. So I think I've spoken in length in the past about how difficult it is to really understand what is executive functioning and what is the value of processing speed across cultures where speed is not really a part of the educational journey. Right? I love telling the story about my childhood where when we were doing tests, we were actually encouraged to sit through. If the test is 4 hours, if you leave early, you would get a worse mark. Like you would be downgraded because why are you rushing? You're supposed to take, if somebody gave you 4 hours, you sit there for 4 hours, even if you're done in half an hour. So it's just an interesting like cultural difference. So I think this is an unsurprising result that Trails B has kind of been, discussed as a very important test of executive functioning, but very problematic in certain cultures where, again, speed is not a component. And also the fact that, Trails B is very, very difficult to adapt to languages that do not use the same alphabet as, for example, like English, Spanish, or even Russian. So in terms of floor effects, so we can see that there is a floor effect in controls. There's also a substantial floor effect in a diagnostically mixed group, and the overall discrimination is acceptable. So what about TAPCAT measures? TAPCAT measures actually perform a little bit better because TAPCAT measures are designed not to have floor or ceiling effects, so the distribution looks much more normal in controls, and the discrimination accuracy is actually fantastic on our test of global executive functioning that we call MATCH, which is a digital symbol paradigm. This is actually the best performing test in terms of discrimination accuracy in this sample and in many other samples that we have examined. So what about language? Language is tricky because there are many different components, and Marlu is here, she won't let me lie, but there are many different things that we have to consider when we develop language tests, and we cannot just presume that by adapting a language test into a different, like let's say, if I ask you, tell me as many vegetables as you can in a minute, right? I'm working with a group in Kenya, and they were asking me, can patients say the vegetables in Swahili and in English? And as a neuropsychologist, my instant response is probably not, right? We want to keep it standardized. We are very, very heavy on standardization. But then I learned that there are many vegetables that are just foreign to Kenya that people have only learned in English, right? So if we're trying to understand the semantic retrieval, if they're able to retrieve what this vegetable is, and they have only learned it in English, why not allow them to say it in English, right? So I think it's just a very interesting, like, cultural development in how we are designing our tests for the future. So again, unsurprisingly, on the test of confrontation naming, the MINT, we do see a significant ceiling effect in controls. This is entirely expected. Confrontation naming tests are not designed to be normally distributed. But I still think it's important for us to consider that as you're thinking about longitudinal data, as people are changing, any test that has a floor or ceiling effect will create problems for you. And then finally, visuospatial tests. Visuospatial tests are very similar to confrontation naming tests in a sense that most people would be probably at ceiling if they're normal, right? You don't necessarily expect people to have profound visuospatial problems, when they're considered to be cognitively normal. But I still think that it is important for us to consider that even participants with dementia have had significant ceiling effects on the Benson copy test, which is a little bit of a concern, which might indicate that this test is not sensitive enough to pick up on early visuospatial impairment. All right, how much time do I have? All right, so this was sort of a pre-intro to introduce you to different types of tests I've been working with, but also the fact that like it's very, very important to do this preliminary work just to look. Data don't lie, so look at the distributions, you know, examine them, see if you can convince yourself that you need to move forward. But now I'm going to move forward to this exciting part of my study that I absolutely love. There is an approach called measurement invariance or equivalence. So the idea behind measuring equivalence is that we cannot really make valid inferences about differences in abilities of persons who come from different groups unless these abilities relate to the observed test results in the same way. In a kind of simple scenario, if two people from different groups have the same score on the test, they should have the same ability, right? That's the idea of invariance, that, you know, if I'm measuring something, if I'm giving somebody a test, they have a 20, I'm giving somebody else a test, they get a 20, they should ideally have the same ability that I'm trying to measure. However, this is very rare. So particularly in cross-cultural research, measurement invariance is fairly rare, and there are many reasons for non-invariance. There are many different biases that go into the overall test development, to the measurement error, like, you know, how used is a participant to the measurement, to the testing environment. There's certain types of item bias, you know, like for example, there's a certain item that just doesn't work well across different cultures. Like if I were to show a picture of a penguin to somebody who has never seen a penguin, this might introduce systemic bias. So those are all important things to consider. I'll give you an example of what measurement invariance is all about. So there's a very widely used instrument called quality of life enjoyment and satisfaction questionnaire. I had to look it up, but it's a very commonly used quality of life tool. And in this tool, participants are asked, during the past week, how satisfied have you been with all of these different aspects of your life, right? And the idea is, when the instrument was developed, you know, the original authors who developed it in the US were like, hey, look, all of these things perfectly align with quality of life. This is fantastic, right? Like, we now have a quality of life measure, we should move forward with this. So the researcher, Gary, in 2022, wanted to see, does this work across countries, right? Quality of life, even when you think about it anecdotally, it's a very kind of subjective experience, right? And when you overlay this with culture, with the economic situation, political situation, it becomes a little bit more complicated. So they administered this to over 1,000 people across these different countries, and then they did a measurement and variance analysis. I'm gonna do a quick poll. Do you think, what items do you think were measurement invariant, meaning that they were the same across countries? Physical health? No? Yes, okay. Mood? Do you think mood was the same, measurement of mood? What about work? How satisfied are you with your work? No? Economic status? No? Okay. Household? You know, your household activities, are you satisfied with those? No? You guys think everything is non-invariant. What about leisure activities? Okay, so the answer is only 2 items were invariant, meaning that only 2 items had the same relationship between the actual question and the overall quality of life, at least in effect or structure, across these different countries. And these items, I was actually surprised, were how satisfied you are with your household activities and how satisfied you are with your leisure activities. You know, so those seem to be the things that we all enjoy together. However, some other things might not be as simple or you know, measurable across different settings. Okay, so this is a little bit of a heavy slide, but I think what I wanted to show is, in order to actually perform a measurement and variance analysis, you kind of go through a series of multi-group confirmatory factor analysis that are increasingly strict in terms of the overall constraints on what the model is. In simple words, it's like you're taking a model that is as relaxed as you let it be. You know, you allow all of your intercepts, all of your slopes, everything to differ. And then in every additional step, you basically try to introduce, well, what if I constrain this? Groups have to be equal on this specific parameter. And then if you establish that, you move on to the next step and you say, now I want the groups to be constrained or equal on 2 parameters or more. And so the idea is that we're trying to understand this overall model, theoretical model that I have in my mind, does it actually stand the test of statistical measurement? Okay, so this is the model that I showed you before, right? These are, this is the actual output from the model. You can see that you can see the names of the tests, which are our observed variables, and our in circles are the latent variables, or the cognitive domains we're trying to measure. And the different numbers there are loadings, the actual, like the relationship between the observed variable and the latent variable. And looking at this, so there are many different ways to kind of estimate model fit, or, you know, like what are the different numbers that statistics tell you, that tell you, is this model working or is this model not working? I can see Renaud nodding. You've seen this before, right? So overall, the model works okay, right? There's one indicator that is not outside of the accepted range, but overall, you never base your model fit decision on a single indicator. So that's why there are many. Overall, everything in green, the model looks okay. Time, okay. I'm gonna move forward, but I will show you that we did test this and we did achieve something called partial strong invariance for this test. What this means is that some of the tests in the battery worked okay and some of the tests did not. So what were the non-invariant tests? What were the tests that were a problem in terms of, you know, comparing Spanish and English speaking participants? The relationship between Benson figure delay and memory was not consistent across cultures. So basically in Spanish speaking participants, when we administer Benson delay copy, we might not be measuring only memory. That relationship was much weaker. Trails A, unsurprisingly, was not exactly related to the speed executive subdomain, and so was line orientation. We also are not exactly tapping into visual spatial skills. And there were a few other items that were contributing to non-equality of the different domains across different cultures. Those were craft story, letter fluency, Trails A, and again, Benson figure copy and line orientation. I think overall, what the study shows is that it's very important for us to use different approaches with the maximum available data so that we can build better batteries. I think I originally wanted to do this because I get asked all the time, you know, I want to do this study in Tanzania. What battery should I use? And I'm like, I don't know. And nobody knows because we haven't really done this before, right? And it is important, I think, for us to harmonize and try to get the studies together in a way that would be comparable. But studies like this really inform how do you design a longitudinal study in places where this has never been done before. So here are a few steps I'm going to do next. I want to look at invariance across educational groups. This will likely be very applicable to our own research programs. I also want to do measurement of variance across different phenotypes. You know, we know that when we test a patient with BVFTD versus patient with Alzheimer's disease, we're probably getting different types of data. And then we're also expanding the study to cross-national comparisons, including samples from Africa, which I'm really excited about. Okay, I'm sorry, I did go over time, but I do want to thank a lot of people, but especially Kate, Victor, and Shireen, and the TAPCAT team and everybody else, Bruce, everybody at GBHI. This has been fantastic. And I will now give the floor to Tanisha.
Tanisha.
Mhm.
Give your full phone.
Okay. I can try to make it as quick as possible. So thanks for the opportunity to share some of the work I've been doing as part of the community outreach team. So today I'll be sharing the proposed project. So we don't have data yet, but hopefully soon. And the project will be focusing on digital storytelling and Afrofuturism and how they can be collectively used to explore barriers Black women experience in the healthcare system. And so before I jump into the what and the how, I think it's important to contextualize this work and our understanding of Black women's experiences within the healthcare system. And so here's a little bit of a timeline. It doesn't detail everything, but we know that present day health is shaped by not only contemporary events, but also historical things, some of which are the things listed here. And so here we see systemic barriers, as well as things like medical experimentation that's impacted Black Americans' trust, some of the things that Charles has mentioned, and biases in particular in how healthcare is delivered. And even it's impacted medical students' perceptions present day. So I think there was a recent study actually that was released that indicated medical students believe Black Americans have thicker skin, which increases their tolerance for pain, right? And so these are things that historically have happened that collectively influence people's experiences within the healthcare system. We also have healthcare policies, some of which are shown here, that have helped and been good as well. So really the point of this is that these things don't happen overnight or in isolation. And so I think this collectively paints a landscape of how there's some medical mistrust, but also a number of ways in which the healthcare system has impacted Black Americans. And so to transition a little bit to cognitive aging in particular, we know that there's race and gender disparities in cognitive aging. And these are actual data from Medicare recipients, so people age 65 and older. And it reflects prevalence rates from 2014 of Alzheimer's disease and related dementias. And so what we see here, you can see my, okay. What you see here is that Black Americans actually have the highest rates, and that Black women disproportionately have higher rates. And they project these estimates to, I think, 2016. What we see is that these disparities actually may grow over time. And so while Black women in particular have disproportionately higher rates, we see that actually they have inequities in diagnosis as well as treatment. And so basically what data has shown is that Black Americans are 35% less likely to receive a diagnosis. And they also tend to present with more advanced symptoms, and more severe symptoms, so later in the disease course. And so what this one study shows is that missing a diagnosis is actually pretty common, but even more common amongst racial and ethnic minorities. And so we know that timely diagnosis in particular is critical for intervention, but it's also, I think about time as a resource that shapes health. And so what we see is that clinical time to diagnosis is actually socially patterned, meaning that access to timely diagnosis differs across different aspects of identity, so race, ethnicity, socioeconomic status. And time inequity is something that I've actually really been interested in. So not only in terms of clinical time, but in terms of time on this earth or lifespan. We also see it in relation to labor. So with Black labor actually being devalued, Black people having to spend more time to make the same amount of money, and then the Black body being commodified. So this dates back to the transatlantic slave trade, where Black bodies were commodified, as well as Black labor. So what we see is that time inequity actually spans multiple domains, including clinical time to diagnosis. And so this time inequity is actually something that's really interesting and has been explored by Rashida Phillips. So she's an Atlantic Fellow for Race Equity, and she looks at it in relation to housing and housing justice in particular. But I think time inequity also impacts clinical diagnosis too. And so what we see is that not only there are disparities in actual diagnosis, but once people enter the healthcare system, we see that there's disparities in access to care. So this is a 2021 survey from the Alzheimer's Association. And what we see is that about 50% of Black Americans report experiencing discrimination when they're seeking care for dementia. And then you can see that people of other racial and ethnic minority groups also report high rates of discrimination. And this is particularly for dementia healthcare. And so also some other statistics, amongst Black Americans, about 1 in 5 people said that they had no barriers when seeking care for Alzheimer's disease, and that only about 50% believe that they could experience culturally competent care and believe that a cure would be distributed fairly. And so when we actually look at caregivers specifically, 42% said they've never faced discrimination when navigating healthcare and feel like they've actually been listened to. And so I think that these numbers reflect a larger problem within the healthcare system and suggest that even for the people that are fortunate to making it to a clinical setting, they're not receiving the care that they need. And so for me, this is where Afrofuturism comes into play, because it's an opportunity for us to consider what the future of healthcare looks like. And so when you think about questions that also have clinical bearing, things like who makes it to the future, who has optimal health in the future, and who has a say-so in what that future looks like, I think Afrofuturism is really important. And so oftentimes people think about it in relationship to like Black Panther or an artistic genre, but it's actually way more than that. So it's an epistemology or a way of knowing. It's a way of generating new knowledge, and it really explores the Black cultural experience and centers Black individuals' needs and their visions. And so in Afrofuturism, Black people are able to create new worlds, basically determining who is seen and how those people are seen. And so not only do they make it to the future, but they have control in shaping that future. And I think it's really important because it's not just really a thought exercise. I know it sounds like, okay, well, what's the point of this? But it's really a way for us to implement like culturally relevant ways of engaging with people to design and basically amplify inequities. And so why is this important? We know that for Black people, having the ability to dream and envision the future is important for people who are unjustly cut from their future, right? So we see this in terms of abbreviated lifespan. We see this in relation to police-related deaths, among a number of other things. So really, placing Black people in the future is a revolutionary act. And I think Afrofuturism is important because it uses a strengths-based approach that kind of divests from this marginalized identity as a deficit narrative, and really affirms and empowers Black Americans and places them as the experts of their own experience. For me, I've been thinking about it in terms of community engagement as well as clinical care, again, using a culturally relevant and resonant way to engage people. And then we also know that Afrofuturism taps into elements of the imagination and the arts, things that we know have benefit for brain health as well as cognitive aging. And so this leads me to the quote of Octavia Butler, who's an Afrofuturist and sci-fi author. This quote really inspired the title of my grant as well as this presentation, because I think the purpose of Afrofuturism is really to bring the things that are unseen to the light, and to bring those people who aren't seen or are part of a conversation to the mainstream and to like a broader public discourse. And so it really speaks to expansiveness of imagination as well as possibility. And so that leads me to my present day work. I was fortunate to receive a grant from the American Psychological Association, as well as funding from Creative Minds. And basically, I developed a workshop series using Afrofuturism for brain health, and it's done in collaboration with the Dr. Davis Center. Sergio mentioned that they're in the Bayview neighborhood, historically Black neighborhood in San Francisco. And so as part of that, we did it over the course of 10 weeks. We did different activities. So some of it was brain health education, arts-based activities, and then also using Afrofuturism as a way to guide conversations around Black women's visions for the future. And so here's some of the activities. So not only were we creating art, but we also had the opportunity to be consumers of art, which I thought was really nice. And we went to a museum. And so over the course of this 10 weeks, I had the opportunity to work with 43 Black women, and I think their ages ranged from anywhere from like 50, and the oldest person was 93 years old. So it was a really nice group, and I really enjoyed that work. And so all of this actually culminated over the 10 weeks in a final photo shoot. And the photo shoot was called The Other Side of Time, and I'll show, if we have time, some pictures from that. And so this was photographed by Austin James. So he's the Hellman artist in residence. And so really fortunate to be able to collaborate with him. And so we also did an exit interview, and it looked like people really enjoyed the experience. And I also was very interested in their hopefulness and their beliefs about their ability to shape the future. And so you can see some of the results there. I also thought it was interesting because some of the women actually came into the project, I guess, with some stigma around aging and their ability to learn new things and engage in the arts. And so some of this, it looks like it's helped to destigmatize those beliefs as well. And so here are some of the photos that we generated. So we worked with 13 women for this, and these aren't all the photos, but some of my favorite ones. And we did interviews with them as well about their visions for the future. And we talked about things like the future of education, future of healthcare, et cetera. And so it was really fun. Okay, so with that in mind, this leads me to my current project, which I'll quickly try to go through. But basically, the first goal will be to examine Black women's experiences of seeking care for cognitive concerns or dementia. And I'll be using digital storytelling, which I'll briefly talk about. The second will be to use that methodology to explore the barriers that Black women specifically experience when seeking healthcare. And I'm interested in looking at it at the hospital level as well as the individual or interpersonal level, so experiences with providers as well as the healthcare system more broadly. And then third, I'm interested in the applications of Afrofuturism so that the Black women can basically tell us what their visions are for the future of healthcare. And so using, again, a culturally relevant framework. And so quickly, what is digital storytelling? It's basically a short film, about 3 to 5 minutes, created by a person that's meant to reflect their lived experience on a topic. It includes them reflecting on their experience as well as how that experience has changed them. And in these reflections, they actually write a script. It's about 250 to about 500 words. That's them narrating their lived experience. And so the experience is paired with archival photos as well as video and music, and then of course their voice. And so it's a process that is collaborative. And so I would be the facilitator working with a group of individuals to create their digital stories. And it's oftentimes focused on a specific theme. So this would be their experiences of the healthcare system. Okay. So there's an evidence base, but I'll quickly go through this. Essentially, people have used this as a research methodology, and they've actually used it for people living with dementia and their caregivers. And it's shown to be pretty beneficial for mood, things like social connectedness. It helps them with memory and recalling past experiences. And it also has been used to create intergenerational connections as well, depending on who's in the group, right? So if there's a topic focused on something that's relevant for older as well as younger adults, then there's that opportunity to have that intergenerational dialogue. For me, I'm interested in using it, again, as a method to explore Black women's experiences. And I think it holds a huge cultural relevance because if you think about Black Americans, we come from an oral history tradition, and Black women are oftentimes thought of as the cultural custodians, so the people that carry on the traditions throughout the lineage. I think it's also important in terms of creating sort of a collective memory of a lived experience. And actually, so in my training that I'm doing, I'll talk a little bit about it, but I've had the opportunity to be a story sharer. So I was a facilitator as well as a person who had to share a story. And I think that it's really healing, like speaking power to your lived experience. So I think there's opportunities for healing there. And then we also know that it provides social and neurocognitive benefits as well. And so I'll quickly go through this, but basically I think for many marginalized communities, being able to engage in storytelling asserts a person's presence and a refusal to be erased from public discourse. And so in terms of the method, I'll be working with people at Story Center. And so I'm actually doing a certification program with them now. And they're actually a collective that comes out of Berkeley, and they're one of the first to pioneer this work as a methodology. And ideally, I'll be working with about 10 to 15 Black women in the community. And it's actually an 8-week process. And so part of it is story writing, but then we participate in story sharing circles where people share their scripts, get collective feedback from the group, go back, revise, and then we go through the process of creating their short film. And so I think, again, in terms of outcomes, I think that there will be psychological as well as cognitive health outcomes. I think that there's definitely power in sharing your stories, and that this can be used to amplify Black women's lived experiences, but also to identify potential areas for intervention. And ideally, I envision this as being something that would be showcased. So showing these short films for the 10 to 15 Black women in a community-based setting. So thank you. Thank you for everyone involved in this work. Sorry it went so long. I think the last thing we have is, Bruce, I don't know if you have any last couple of words.
How can I say anything after that? Wow. Thank you, Tanisha, Elena. What a beautiful day. I think even though Elena was brought up in a place where she was taught to be slow, she's overcome it for sure. Okay. I think we have time for a question or two. So I might start with Elena. I'll ask a question. And it seems like we're far away from good tests for our red lat cohort in South America.
Great.
Does HABCAT work for executive, spatial, memory?
Is spatial.
Yeah.
I did not anticipate this at all, but I think the fact that visual spatial measures are so difficult